Welcome everyone to our workshop on open science. Namely, we would like to discuss the importance of making science accessible to the general public and giving other scientists the tools required for proper replication and transparency. This workshop will involve 3 quick presentations, and then we will end with a Q&A session for all 3 panelists. So please hold your questions until all 3 panelists have finished their presentations. So let's begin with the presentations. The first panelist for this workshop is Justine Hansen, a PhD student in the lab of Bratislava Music at McGill. Justine has spearheaded 2 promising open science projects. In her first project, Hansen and an international team of researchers mapped a brain receptor atlas to neural structure and function and published the results in an open preprint. She also made the data and code openly available on GitHub. Her second project, Neuromaps, uses more than 40 existing brain maps to help scientists find correlations between different brain regions, spatial scales, modalities, and brain functions. The software required to replicate these analyses was also made publicly available. For her work on these 2 projects, Justine was awarded a 2022 Neuro Irv and Helga Cooper Foundation Open Science Prize. Today, Justine will be presenting her open science journey and will speak about the big picture of open science. The floor is yours, Justine.
Works now? Yeah? Okay, cool. So hi everyone. So I've titled this Who Am I and Why Am I Talking About Open Science? Because it's a very fair question. So first I'll start with the open science bit. I Googled this to see like what is open science. And the government of Canada has this like nice infographic. And as you can see, it's like a lot of things. So open science is really multifaceted. It can be kind of overwhelming to try to dive into. It has aspects starting from ideation. So that's that point 1 here, things like in science, I don't know if you guys feel this, but we can be a little bit closed about our project ideas because some people are afraid of other people scooping them or stealing their ideas or whatnot. Open science spirit is the opposite of that. It's really to keep things open, to let other people influence how you are going to approach your project and whatnot. The second part of open science is maybe what we're more familiar with, this kind of data collection and analysis, making your code open, making your methods open, making your data open. Third part, publication. Maybe we don't think about this as much, but of course it is also important to make your publications, once you write your paper, open to the general public, not just the scientists. So, I mean, we often have access to a lot of the scientific publishing material out there because McGill or University of Montreal, et cetera, they're paying for those subscription fees. And so we have access to them, but most people don't have access to these things. And it's not very helpful if all of our science is only available to other scientists. Part of the point is to make our science open also to the general public. And that's where the fourth one comes in, this knowledge spreading or communication. That's really about being able to talk about your work to general audiences, to specialized audiences, writing a tweet print or whatnot, having a Twitter thread about your work and making it more accessible to other people. There's this also really nice, do you guys see the Zoom thing? Okay, cool. There's this nice Turing Way path. They have a lot of cool illustrations. I suggest you check it out about reproducibility and open science and whatnot. But here's also showing you that there's so many points to getting into open science. And because of that, it can be very overwhelming. So I want to emphasize that although there are so many ways to be involved in this open science movement, you don't need to be expert at all of them. I am certainly not an expert at all of them. And so that's where I go into the why am I here part. So I won an open science award. That's why I got invited. So they're, the organizers, thank you very much for inviting me. They were like, hey, you won this award, so you probably know a lot about open science. Do you want to come talk to people about open science? And what they didn't know and what I didn't tell them, so I said, yes, of course I'll come, and then did not tell them. The whole thing was kind of an accident. So I did not start thinking I'm an open science queen. I'm going to go in and open science, and I'm going to win this award. It's going to be great. I'm going to be invited to give talks. I was thinking I'm going to do my PhD project. Oh, look, some stuff was open. Oh, look, I won this award. How great. Oh, now I'm being asked to talk about it. And people think I'm an expert about this. Obviously, it worked out well for me. I mean, hopefully as long as nobody finds out I'm an imposter. But for now, it's working out really well. And so my message to you, first of all, I'm still early on my open science journey, but also my message to you is that you don't have to be an expert to get started, which is very obvious. Like, of course, you don't have to be an expert to get started, but sometimes it can be very overwhelming. And it can feel like you need to be able to be good at writing really amazing software and code, and you need to know how to use GitHub and Git, and you need to, pay massive open access fees for journals to publish your work. Open access, that's a totally other thing that I'm not going to talk about right now. But you don't need to be pros at all of this. You can start by implementing some small habits, and hopefully that will get the ball rolling and you'll be more involved in this kind of open science movement. So I'm going to talk just very briefly about the projects that kind of got me into this open science movement, and through them explain how it was all an accident. The first one is about this project mapping neurotransmitter receptor densities to brain structure and function. And of course, receptors, they're so important in, literally action potentials firing, right? So neural dynamics, probably brain function, but we don't know very much about them at the whole brain level. And part of that is that we don't have the data. So I was interested in mapping these receptors to structure and function, but I didn't have the data. So I started asking people first at the McGill community. So here I'm showing at the bottom left, the institutions and the people in the right side, is that the right side? Yes, right side. The people that I contacted, and I said, hey, you guys are pet researchers, you have this data, would you be willing to share this with me? And by the way, would you be okay with me sharing the group averages on GitHub afterwards? And everyone was super positive. So we started local, we started branching out a little bit. We went eventually to other provinces. We made it to Finland, so that's on the other side of the ocean. We made it to Yale. I don't know why we first went to Finland and then across the border, but we did. Also a bunch of stuff in the UK and Switzerland. And there was some data that was actually already open. So this is data from Denmark and also from Germany and Jülich. And you can see this list of people, all the people that contributed. And a lot of this data was previously closed. And it wasn't closed because they wanted it to be closed. It was closed because they weren't really thinking about open science. Nobody had asked them to share before. They didn't really necessarily know where to put it. All it took was me saying, hey, I would like to work on this project. Would you be cool with sharing your data with me? And by the way, is it okay if I make it open? And everyone was very positive about that. So this goes to show that you can be very research forward, focused on your research project, but then kind of by accident put together an atlas that now this is 19 receptors and transporters. It's 9 different neurotransmitter systems across more than 1,000 individuals. It's an open atlas. It's already been used a lot. And now people think I'm an open science guru for doing this, but it was kind of an accident. Okay, project number 2. This is a toolbox. It's a little bit more open science forward, but here neuromaps is what it's called. It's a toolbox for contextualizing brain maps, and what it does is it helps us make transformations between different data types in neuroimaging. So if you're not a neuroimager, don't worry about it. But in neuroimaging, the brain maps can be available in different ways, and so it can be convenient to be able to convert these to each other, to compare them to a bunch of data in the literature, to make robust statistical inferences about these, and then with the goal of being able to enrich your analysis, get an idea of, is your fMRI task activation map related to gene expression, receptor density, cell types, neural dynamics, etc. But one of the key things about this, and by the way, that's Ross, he's really the muscle behind a lot of neuromaps. One of the key things here is standardizing workflows. The reason neuromaps was built originally is that we in the lab all realized we were doing really similar analytic pipelines and workflows, but we were all kind of doing them by ourselves in terminal, or in a notepad or something really stupid. And we were like, well, if we're all doing the same thing, maybe we should make this standardized. And if it's useful to us, maybe it'll be useful to other people. And hopefully that will make these analytic pipelines a little bit more reproducible, because it'll give us an idea of what exactly is being done, and it's all open so people can see what it is that we are doing and how we're doing it. Okay, so main point here is if you're already really into open science, that's awesome. If you're not so much into open science or don't think you are, then I'm just going to give you a few pointers for how to integrate some habits into your master's or your PhD, your research. Put in a little dose of open science to try to get the ball rolling. So one thing is document or comment your code. Do this for your own sake. Don't do this for other people. Just 2 years later, when you're let me do that analysis again, and then you go back to your really messy code that's in tmp.py or something, do yourself a favor and keep things documented. It'll make things easier for yourself and also for other people who are reading your code. Track changes. This is controversial because then you have to learn how to use Git, but if you feel like taking that step, I would recommend it. These 2 things, documenting your code and tracking changes, it'll make it easier to make this public GitHub repo. So if you have your code organized, it's not very difficult to just put that online. And the reason I originally put my code online was because somebody else in the lab was doing it. So I was like, sure, I guess I'll do it too. And then people started sending me emails. Hey, I was doing this analysis. Can you tell me about what you would, how would you do it for this kind of data set? Or how would you change it for this? And I was like, oh my gosh, people are actually using this. So I think it can be very useful. Put up a preprint. That's very easy unless you have 100 authors on your paper. So just do it. You get citations, if you want to be selfish. I mean, obviously we all want to be selfish, but maybe not obviously, I should have said that. But put up a preprint, you know, it's useful and it's open. Everyone can read it. Tutorials are a thing, so you can do a thread on Twitter and hopefully get your work to access a broader audience. Make as much open data as possible. I put an asterisk here to remind myself to say, of course, there are ethical requirements, so you shouldn't be making anything open that's not yours to make open. Talk to the people who actually collected the data if it wasn't you, etc. Make sure like you're not sharing individual data, that's a problem. Notebooks are a thing. So if you make, for example, like a Jupyter notebook, make it possible for people to run your code directly. That makes it really easy to make your work open, but also reproducible. And lastly, communicate your work. So give talks to scientific audience, give talks to general audiences, write a blog post if that's your thing. If you're social media savvy, there's like, do a whole thing there too. These are kind of ways to implement open science in your research, but you can keep it research first. Like you don't have to commit yourself to like the open science gods. You can just do your research as you otherwise would have done that and sprinkle in these little habits, and that'll kind of get us rolling in terms of trying to make science in general more open. Okay, that's all. I'll hand it off to, no questions. You may not ask questions. I'll hand it off to the next speaker.
Thank you, Justine. Our second panelist is Yann Harel, a PhD student in neuropsychology and cognitive sciences at the Université de Montréal. He joined the Computational and Cognitive Neuroscience Lab in 2016 as a student researcher supervised by Professor Karim Jerbi. As a member of the Quarton Neuromod project team led by Professor Pierre Bellec, he has also been involved since 2018 in the creation of an openly accessible multimodal brain data set with the aim of stimulating research on machine learning. Today, Yann will be presenting his background and will be showing us the core tool for version control using GitHub. The floor is yours.
Hi, everyone. My name is Yann, and today I would like to take maybe a bit of a pragmatic approach of open science. I remember being a student and hearing a lot about open science, but never really knowing like how to contribute, how to add my own piece to that. And in the end, I actually realized that open science was maybe more of a mindset that was reliant on common tools that are not necessarily open per se, but that will improve your workflow and process and help you make your science more open. So that's kind of my point today. And to do that, I wanted to start again with a simple reminder of what we are doing here. What is the scientific method? And I found on the internet, I found that guy who had a pretty simple explanation for what we are doing. I like it because it's not going into consideration of like what should be the perfect scientific method, or is there are plenty of ways to do science, but in the end it's always about the same thing, which is I'm gonna say messing around and finding out. And the more you mess around, the more you find out. But the thing is, when you go into that process, you end up doing all sorts of things. You end up with a lot of code. You end up with data. You end up with a paper, hopefully at the end. And if that's not enough, and you want to keep going and do the things correctly, the right way, you will also need to document your code, and you would maybe want to share your data with everyone else. And so to add the openness to that process, I think the most important part, which is also like fundamental for science, that would be writing it down correctly and keeping track of everything that happened in the process. The product of your scientific inquiry is not it's not the paper, it's all you did to get there. And that's what I want you to document. And I wanted to show you a few tools that I'm currently using to do that. And these tools, so most of you are probably already familiar with GitHub, but for the very few of you that are not, I still wanted to make this an important part of this presentation. And I will also tell you very quickly about BEADS. So it's a standard system for brain imaging, and show you a few data sets that are using these tools. So basically, at the end of this talk, you would be able to go out there, find an open data set that you'd like to work on, and just download it and start playing with it. So let's start with GitHub. Why are we talking about Git, and what's the point? Well, all of you are students, so I assume that most of you, if not all of you, have had this problem before, where you write something, then you get corrections, then you modify the document, and then you make another version and another version and another version. But at some point, your advisor is like, "Oh, but I remember I like that paragraph you had 2 versions ago." And you're like, "Oh my, no, I cannot do that. Like, I don't know where it's at." Well, basically, GitHub Git is a solution for that. The reason I say just Git and not GitHub is because it's 2 separate tools. Git is actually a command line version control tool, and GitHub is a web hosting of the repositories that you will create using Git. But basically, Git is something that runs locally on your machine, and that helps you keep track of the successive versions of your code or text or whatever. And GitHub helps you share that with the world. And also, it's very convenient when you work on different machines and you want to synchronize your code across the different computers, for example. And the way it works, there is this simple, like a small graph where you see it's some kind of tree where you have a trunk, and you can create branches which are variants of your trunk that will keep evolving on their own until you merge them back to the tree. And the reason why using that tool is very important is because if multiple people are working on the same project, which is often the case in open science, well, they can still work on their own version of the project without messing with whatever the others will need. And then the tool will help you merge the different versions and solve the conflicts. But since we are students, we don't have a complex use of that tool. And what I would like you would be to adopt a very minimal and simple use of Git, which is summarized in this comic. Basically, just type a bunch of commands. Anytime you have a working code session, you're like, "Oh, I want to fix that bug." You work 1 hour on it, then you synchronize. And I want that to become a reflex. That's 95% of my Git usage is just doing that. But just by automatizing this process, you will create actually an historic of how you code, how your code evolved to become what it is now. And these commands, these commands are really simple. You have the git add command, to add a file or a folder to your incoming commit. You have the git commit, which will actually take all the files you've added recently and create an historic for these files. And then that's also an important moment to document what you are doing. So this is where you are going to explain the change you just made. And then you git push, which actually synchronizes your folder with GitHub, for example, or GitLab. And I also wanted to show that for some of you, it's a screenshot of my Visual Studio Code environment. And for some of you who are not very at ease with command line interfaces, well, you can find integrations of these tools in most development environments you will have. So in VS Code, it's a simple, yeah, you can see the cursor. So basically, just in that part of the screen, you just have to click the plus here, write a little message in the text box, and click commit. So in order to go deeper on Git, I would definitely recommend that you go there, find a repo that you like, and try to play with it, fork it. So that means copy it to your own account, modify it, try to push it. And as I said, 95% of my Git usage is very simple, and it's what I just showed you. But the 5% that are left is what I'm trying to work on. I'm trying to use Git as much as possible for as many applications as possible. So communicating with my team, for example, about an issue I found in a data set that we are using is something that I'm going to do through Git, because first it will be traceable, but also it centralizes all the information, like all the issues of on a single project, and yeah, it just makes your workflow more how do you say, more flexible and more efficient, really. And I will encourage you to go see these other tutorials that will give you a more complete tour of the different usage you can have of Git, and you can. Yeah, as you remark, you can also host a website using Git, so that's still a plus. Now I wanted very shortly to talk about DataLad, which is the equivalent of Git, but this time for data, because Git is only it's only designed to track code and other types of documents, but it's not suitable for data. For data, you will be using DataLad. It's a bit more difficult to set up, so I don't expect you to go back home and push your data using DataLad, but at least know that this tool exists, and if you find an open data set, it will probably be tracked using DataLad. And the commands to use DataLad are very similar. It's the same concept as Git. Actually, it's built on Git. So now another aspect I wanted to tell you is about how to standardize your data. And for that, I won't leave many options on the table. We are doing neuroimaging. And I think one of the most flexible framework you can have for standardizing your neuroimaging files will be the BIDS system. And basically, BIDS, it's just a set of rules that you can decide to apply or not when you choose a name for your files. It's just a structure where you have your data set. Every subject is called sub01, sub02, sub03. There is a TSV files to summarize metadata about your subject and your tasks and everything. And in each subject folder, you have different folders for different imaging modalities and so on and so forth. But basically, BIDS is one paper that you can find here that describes all the rules, and it's also a set of toolboxes or softwares that are compliant to these BID requirements. So whatever the imaging modality you're working with, it might be on this list. If it's not, it will be fairly easy for to actually create a plugin to manage this data. But in most cases, you won't even have to. And also, formatting your data in BIDS can allow you to use some more advanced tools. I'm thinking of M&E BIDS pipeline, which is a pipeline that takes your data from the pre-process, like the raw stage, pre-process them, extract features, run some analysis and classification. But you can only do that if they are properly formatted in BIDS. And finally, I just wanted to touch a word on open datasets. So the one we're working on is currently called Courtois Neuromod. It's a longitudinal dataset that you can find on the cneuromod.ca page. And we have plenty of tasks, video game tasks, movie watching, story listening, and also a reproduction of the HCP protocol. And if you want more varied data sets, you can also go find more on Open Neuro, which has more than 800 public data sets on various imaging modalities. And all of these data sets, Courtois Neuromod, the ones on Open Neuro, they will be BIDS compliant. They will be hosted on DataLab, and you will probably find GitHub repositories that will provide you with a bit of code to play with it. So that's it for me. And if you have any questions, save them for the round table. Thanks.
Thank you so much for your wonderful talk, Yann. Our final panelist, Dr. Hao-Ting Wang, is a postdoctoral researcher in the Laboratory for Brain Stimulation and Exploration at Montreal Geriatrics Institute, working with Professor Pierre Bellec. Dr. Wang's current research focuses on brain connectivity-based biomarker discovery through machine learning. Her open science contributions include the neuroscience and machine learning software NiLearn, CCA Zoo, Prada Workflow Task, Prada FSL. Dr. Wang recently hosted the OHBM Hackathon 2022 in Glasgow as the OHBM Open Science Special Interest Group Hackathon co-chair. In this talk, Hao-Ting Wang will talk about how to best contribute to the open science community by starting a package or contributing to a project.
All right. Thanks for the great introduction. You did a very thorough research on me. I didn't expect all that. So today I'm going to finish this talk with talking about open source projects specifically, and open source projects and how to find them. Is it this computer? All right. Okay. So my day-to-day now is more like a software developer, but my background is actually psychology. And there are two very important phases in my journey to open source software. So I started as a PhD student who just shared my code online through Git. I was thinking I was doing pretty well, but it's pretty incomprehensive code, really. And then after some, well, I don't know, soul searching, I become slightly more reproducible with my practice when it comes to research code. And just to give you an idea of what kind of person I was when I was a PhD student, I really don't like Wi-Fi, and I insist on using landline to connect to anything. And I was already a Linux user at that point, and I have a inflated sense of self because I can code in Python and I can use a terminal. So what happened? What happened is like towards the end of my PhD and made me a slightly different person. So at that time, in my lab, there's a side project I was helping on that needs to use cortical surface data. And there are some problems here. The cortical surface data is not very representative in the Python FMI community. And there is a general lack of tools to read and save the data. And at that time, I made a solution that doesn't require any extra dependency and can just read and load data really, really well. And I think this is a good solution. I want more people to use it. However, there's a reality. This is a temporary code I wrote for my lab as a side project. I will gain no credits in creating this piece of work and making a software contribution. What did I do in the end? I ignored my PhD papers and I submitted a pull request to Nibabo. That's what happened. And throughout this whole process, I learned some very, very valuable lessons and made great friends along the way, which is a lot of these open source softwares are maintained by people who really, really are not paid to do this. A lot of major these neuroimaging infrastructure that maintained by unpaid volunteers. And the best way to make them feel appreciated is actually contributing and be part of the community. So since this talk is mainly targeting towards students, I'm going to talk a little bit about me as a maintainer, software developer. What do I do if I'm a user, like students, to contribute to open source software? And lastly, I will address a little bit of how I feel as a maintainer. So start from being a user. A user's job is to choose the tool to use in your own research code. And choosing a tool sounds simple because a lot of us are already doing scientific research. A lot of the tools we are using are already open source. And there are going to be some tools that you're going to be using that comes from very specific lab and they have very specific algorithms. And sometimes you will find those code unmaintained. So here are some tips to help you to identify if you have multiple choices of these small projects, how are you going to identify the more reliable ones to use? So the first thing I will look at is the level of activity of that project. So I give you two examples from bigger softwares over here. So first one is Nibabo. It's a software to read and write neuroimaging data file formats. So it's supposed to be very, very stable, but it is still having a very active activity. It is still releasing once in a while. And the bottom example is Nylon. And in this example, we can see people are already, they posted an issue 8 hours ago, and there was already someone working on it. It really shows that people have been investing time into this piece of work. It's actively being maintained. And the next thing I will introduce you to is to look at whether the project has tests. This is a surprisingly uncommon thing in scientific software, especially the ones that you find that has a peer-reviewed journal paper published accompanying it. You don't see tests, but that is a very important factor to understand if this piece of work is actually robust and worked on by an engineer or a developer. And since over here we have some people from the AI background, and there's another extra things I will introduce to look at, which is in the source code, do they use assert to catch errors? If they use assert to catch errors in the code that working in the analyses, this is a really, really warning sign I would like you to get rid of. And we can talk about that in Q&A or ask me over coffee, really. I'm not going into detail to this. So you wrote some code and you want to contribute. But contributing is really not that easy when you're talking about contributing to a piece of software, because there's a lot of extra tools you will need to learn before you really can feel comfortable contributing it. And by my own experience, I can tell you, people will help you even if you don't have these skills. But it's going to make the experience a lot more smoother if you have those skills. And then we talk about sharing code a lot, but sharing code is easy, but sharing code properly is hard. And we have so many people just share their code and it is not readable, and it's not reproducible because data is not annotated correctly. So I'm going to give you 2 tips to how to overcome this at your own personal capacity. So how do you contribute without a PR to a software? So first middle point is, instead of sharing code, think about sharing comprehensible code. And there are several elements. So firstly, I want you to look at the dependency of your software. Most of the time when I'm executing other people's projects, the first thing I'm going to solve, and the majority of the time I'm spending on, is to figure out their dependency. If your dependencies is reproducible on any machine, then chances are you can use other services like Binder Hub to reproduce your analysis really easily. And document your code. Please use standardized format to document your code. And I will introduce you to the NumPy style guide. You can look it up later. The reason is they have a standard list of things that you need to specify while you're documenting the code. And that will really help you to understand what is missing in your current project. And then the last 2 things are optional, but they make the code a lot readable. So first thing is code style. And here I give you a few tools that will do some code style for you automatically, like linter to find out if the syntax is like a standard syntax, code spell to check your spelling. That is quite common. And sometimes I found my own bugs due to spelling errors. And code formatter, it just makes it a lot easier to read. And now there's a one-liner tool to help you do that. And finally is a code quality. This one is a very difficult one to crack. But if you want to improve your code quality, I would recommend you to use things like GitHub Copilot as a paid service. And then there's another project I use. It's called Sorcery. And it is a plugin that you can install on some IDEs, for example, VS Code we are seeing today. Another middle point is to try to learn to write small applications. The very simple principle we are looking at here is just to make each program do one thing. And here, this is a workflow from a previous piece of work that I've done. We have multiple steps to generate some benchmark results and release our data and our figures to NeuroLibre. And at each part of that, each of these graphs in this workflow, they can be containerized into an independent software. So when I was writing the project, it was a pile of code. But later on, when I was trying to actually make this thing a usable tool for our lab, for each of those steps, they are a small application. And you can practice a lot of software engineering practice if you wish, if you reduce the code to a very small chunk. Another benefit in scientific lab to do this is the code base is small. So it is easier for other people to learn. It's easier for knowledge transfer. So finally, we don't have that much time left, but I'll talk a little bit about being a maintainer. And when I was writing this part of the talk, I was thinking, do I really want to talk about this? Because as people has introduced me already, and you haven't probably noticed, I don't own any software. There's no software under my name. Why? I made an iceberg chart over here. I don't expect you to actually read it. I just want you to know there is a huge amount of overhead once you make a software package and once you're responsible for a piece of software. When you have the ability to make a package, chances are you have the ability to make a contribution to existing tool as well. And I would encourage you to do that because that will take off a lot of uncertainties for the future of this piece of software. And if you want to make sure your tool is benefiting the scientific community, I encourage you to think about not making a package release like that easily. So finally, here are a bunch of links with projects that I talked about that can be used as an example. And the first 2 resources is the 2 resources I think they are the most important one. So the first one is telling you how do you set up a data science project in a Pythonic way. And the second link is a lot of tutorial for Brain Hack School. We have tons of tutorials covering the skills that I covered today in the talk. Yeah, thank you very much.
Thank you very much for those 3 amazing talks. Hao-Ting, you can directly stay on stage and take a seat here. And I would like to ask our 2 other speakers and Mel, who is my co-moderator for today, also on stage for the panel discussion on open science. It will be about 10-15 minutes long, so we're not too late for the lunch break. So my name is Charlotte Maschke. I'm the chair of the UNIC Student Affairs Committee and moderating the open science panel today. During the open science panel, we are starting with just some kickoff questions that we've prepared, but we invite you, of course, to ask questions. There's a mic installed over there. There's a mic installed over there. So if you have a question, just feel free to navigate yourself to the mic and ask a question. Should I start with, you will start with the first one.
I guess I'll start. The micro works, so that's good. So I think we have, we get some idea from the 3 of you, but maybe if you can summarize in 1 or 2 phrases, what is open science for you? What do you what do it means basically for you to work with open science? Yeah.
I can start. So yeah, that's an interesting question, and I think each of us tried to kind of answer it succinctly during our talks. But my view is very I don't know. As I said, without going into epistemological considerations, when you do science and you document, part of the science you do is documenting it and writing, telling a story with what you just did. Well, if that story is something that you give for free, it's something that that's accessible. You made it available for as many people as possible. You made your code accessible, your data, everything is reproducible. Then that becomes open science. And if your story, if your documentation has some embargo on it, and some people can read it, some others cannot, well, I would call that closed science. So basically, I don't think open science is different in nature than regular science. It's just a mindset that you want what you produce to be available to as many people as possible.
I simply don't use that term. So the reason is actually because this term is a really loaded term at this point. It involves the diversity, it involves data sharing, it involves code, and it involves publishing practices. And this is not a single skill people can master. This is not a single set of skill. People might be more specialized in one of the aspects. So when I talk about open science, I will usually talk about what is my actual expertise. And my expertise is contributing to open source software. So I will usually ask people to clarify, what does it mean to you? Because that way I will be able to answer the question better. Yeah.
Yeah, I really like, I think Yann said this in his talk. He kind of said it's like a mindset, which I totally agree with. It's not so much what you do, it's just how you're thinking. Are you trying to be honest about your work? Are you trying to be transparent about your work? If somebody asks you the project that you're working on, is it a secret because you're worried about being scooped, or are you open about what it is that you're working on? So that's my short answer.
Thank you so much. There's a question from the audience.
Hey guys. I have a question because all 3 of you, I happen to know, come from labs that are really pro open science. And it would be interesting to speak on the challenges. Not everybody possibly in this room comes from labs that open science is a culture. And so maybe talk about some of the challenges of what it might be like and how to overcome those in a lab that's maybe not so open, or an institution that's not so open.
Can start with that. So before I joined the Courtois Neuromap project, I was working. I still work with the COCO lab with Dr. Karim Gerbi, and I started doing my first thesis experiment in that lab. And I wasn't, of course, I was like interested by open science. I wanted to do things as openly as possible. But I pretty much wanted just my project to get started. So I've asked for an ethics approval to recruit participants and to scan them using our MEG system. And at some point I was like, oh, nice. I have this great data set of nearly 30 people doing some sustained attention task in a MEG scanner. And then I'm going to have some anatomical MRI data to do source reconstruction. That data set is interesting. And I know like there are plenty of analysis. I spent countless years thinking about the different analysis I could do on this data set. I won't do them all, that's for sure. And if you guys want to do them, I would be glad that you take interest in this. But actually, I can't. I can't because this ethics approval doesn't cover the fact that I would share the data openly. So I didn't ask for my participants' consent about that specifically. So I would still be able to use that data if we were to collaborate on something. But I cannot just put my data on Open Neuro, for example. And that became a bit of a frustration to me. Now I know that I would have loved to do the things differently. But talking with my supervisor also helped me make peace about that, because now that I've seen all the efforts that the Courtois Neuromod team made to get their data available, I'm not even sure that I would have been able to maybe it's too much work and too much effort for me as a PhD student who just want to get started, who has all the freedom I want, but it's still a lot of efforts to get through ethics, to get through paperwork, and because you want to do the things right, you know, it's a sensitive matter. You don't want to take it lightly, or at least not as lightly as I would have loved to. And that's why I think we are very lucky to have Hao-Ting here, reminding us of, yeah, it's cool, it's all nice talk and everything, but then there's hard work involved, and it's a tedious process. It's not always like you can spend 100 hours making your data set available to just have no one download it. And is it worth it? Yeah, but
Yeah, well, we are very, very lucky because I come from a very interesting position, because throughout my whole PhD, I used two open access data set for my PhD. And then I joined a lab that doesn't really have that much of open access data sharing practice. And I would say if you are in that kind of environment, what you need to do is something that is within your control. And that's what I try to say in my talk today, because a lot of things I'm talking about today, for example, just documenting your code in a more accessible way, that is going to be very beneficial to your future PhD coworkers, even within a lab that doesn't encourage those kind of practice. So you need to focus on what you can do within your control. It is very tempting to talk about all the exciting stuff, sharing your data and all that, but sometimes it really starts from the very small groundwork. Yeah.
Yeah, I guess my perspective of this is that a lot can be done just by leading by example. So to give you an example, my supervisor, Bratislav Misik, he's certainly now known kind of as an open science proponent, and he has a lot of momentum there. But that's not how it started. I give a lot of credit to Ross Markella, who is a PhD student who has now graduated. I put his face up on the slide during my talk. So he's graduated, but when he joined, he was very focused on open science. And he was kind of doing a lot of these open science practices on his own, just because he cared about it and wanted to do that. And by doing so, Bratislav realized how much of a benefit it was and how important it was. And so now Bratislava is an open science proponent, and now it's kind of a cultural in the lab that we all participate in these, you know, we try to have a GitHub repo and whatnot. So if you're interested in this and your PI isn't, I would recommend that you try to move in that direction and kind of show your PI, hopefully, as if they're not, you know, shutting you down, but show your PI how beneficial it really is. I think they will notice that it's a good thing and then try to implement that for the rest of their students.
Thank you. Maybe a follow-up on this question, because the question, like, that's also one of the questions that we've prepared, because it's super relevant for most of us, being in a position where maybe we're working with data that exists in the lab already and we don't have the right to share it. But it's just important to say that there are so many other ways to start in open science. Even if you cannot share the data, there's still so many things you can contribute to. So maybe I just wanted to take a moment to talk about preprints, what they are and why people should use them. I feel like in my studies, I've learned about them way too late. So maybe there's some people in the audience who still don't know what it is, and it might be a good moment to just introduce preprints and talk about its advantages.
Okay, I'll try to make it quick. So pretty much a preprint repository, it's free, it's accessible to everybody. Your work has not yet been peer reviewed. I highly recommend that if you're going to put your paper that you've typed up into a manuscript on a preprint repo, do it when your paper is ready to be submitted, because what's good about preprints is that you can, it's not peer reviewed, right? This is important, but it shouldn't be like a kind of half-assed, like semi work in progress. It should be something that is you're ready to submit to a journal. It's really beneficial because it just means that your work gets out there earlier. And Google Scholar, for example, can track your citations. It'll track when a preprint gets published, and so you'll start getting cited earlier. Of course, if you're reading preprints, which I highly recommend, a lot of the work I read it at the preprint level. The grain of salt is that it hasn't been peer reviewed yet. So you just need to you should always be critical and you should always be, thinking about this, but yeah.
Yep, I'm using this opportunity to advertise a new preprint service called NeuroLibre. So in this project, you can upload your code and make other people run your code and even some of the metadata. And the skill you will need to require is actually also covered in my code talk today. You need to learn how to build a dependency tree that is usable through services like Binder Hub. And right now, the platform is still under construction, but I hope it will be available to people very, very soon. So yeah, consider NeuroLibre in a few months' time.
I won't I'm not sure about the advantages of preprints over regular papers, but I'm mostly doubtful about the advantages of regular journals over preprints. I know that's like the peer review system is something that's very fundamental for science, and it seems that with tools like NeuroLibre, PsyArchive, bioRxiv, and so on, we have a great tool to share our work without being too much constrained by the requirements of the academic publishing system. So just for that, I would encourage you to use it as much as possible. And then, I think getting your paper peer reviewed is something really crucial. But if you can just put a preprint in the meantime, it doesn't hurt anybody. And I think that's the most important part, is that your work is out there, and that the review, the fact it's reviewed or not, is transparent. And from there, it's just free lunch, I would say.
Okay, maybe just one quick comment, because then we need to wrap up already. But you want to add one comment?
Okay.
Thanks. Yeah, I just wanted to, because you reminded me, when you have a preprint out, you get your peer review kind of in an informal setting very early on before you do the official peer reviews. Yeah, so that's really nice that when you put it out there, you can get comments and people will tell you, what about this? They'll ask questions or they'll give comments. And some of the time, if not a lot of the time, you'll say, wait, that was a really good idea. Let me implement that. and then submit that better version to the journal and hope it gets accepted.
Okay, so as you've seen in this really quick roundtable discussion, this discussion could continue for a long, long time. And I hope it will continue like among you, with other students, maybe even with your supervisor, just bring it up in a lab meeting, because it's an extremely important discussion. And beside this, I would just invite you now to continue this discussion with us also over lunch. It's now time for the lunch break, which will take until 1 p.m. So I would like to invite you back to be here in this room at 1 p.m. And the lunch is not in front of this room. The lunch is actually in the atrium. Just follow the signs or follow the crowd. We will go to the atrium together and then find ourselves back here at 1 p.m. for the next session of this conference. Thank you so much.
Thank you, everyone.
And thank you to our panelists.